So, dear colleagues, I would like to start with a short introduction. So my talk will be devoted to the fragmentation of the myelin basic protein by the proteasome without the involvement of the ubiquitin. So just to remind you that multiple sclerosis is a chronic neurodegenerative disease of the autoimmune background. And the myelin basic protein is considered as one of the major autoantigens in this disease. And in turn, these fragments of this protein are targeted by the cytotoxic lymphocytes that release the oligodendrocytes, and this leads to the axonal loss. So the proteasome, or 26S proteasome, is a major supplier of the peptides generated from the endogenous proteins that through the TAP-dependent mechanism are presented on the MHC class I molecules and these peptides are recognized by the T cell receptors of the cytotoxic lymphocytes and this some kind of the molecular passport of the cell that helps the cytotoxic lymphocytes to recognize some pathogenic or transformed cells. There is a very difficult and evolutionary perfect system called ubiquitination system, and consists of more than 700 members, and this is cascade of the ubiquitin ligases from E1, E2 and E3, and each protein to be hydrolyzed by the proteasome should be ubiquitinated. And so the current dogma is that if the protein is not ubiquitinated, it feels himself quite safely near the proteasome because it will not recognize it. And we asked several questions, starting from this one. So why oligodendrocytes do not use this highly evolved system to control proteasome-mediated hydrolysis of the myelin basic protein, and therefore being defended from the cytotoxic lymphocytes. So there are three main questions. The first is the myelin basic protein as a proteasomal substrate. The second is proteasome-mediated MBP hydrolysis somehow involved in the MS progression, and may proteasome be a target for some therapeutic intervention. So first, we showed that the myelin basic protein is a proteasomal substrate. You see here that during the cyclohexamine chase, the amount of the myelin basic protein is decreased in these cells, and when we add some specific inhibitor of the proteasome, PS341, it is stabilized. Now, what was really interesting that when we precipitated the myelin basic protein from the cells, you see it here, so we did not detect any trace of the polyubiquitination in contrast to the positive control P105. So the conclusion is that the myelin basic protein is hydrolyzed by the proteasome. Nonetheless, it is not ubiquitinated at all. Second, what we tested, the influence, the effect of the siRNA targeting E1. So we completely block the ubiquitination. And the second, we use so-called ubiquitin K0, the variant of the ubiquitin that lost the possibility to form the polyubiquitin chains. And as you can see, the degradation of the myelin basic protein was not changed. It was hydrolyzed in both cases, in case of ubiquitin K0 and in case of addition of E1 siRNA. In contrast to the classical ubiquitin-dependent substrate MYC, it was stabilized in case of ubiquitin K0, and same for E1 silencing. So the next suggestion was that probably MBP has some direct proteasomal signal. And to to test this hypothesis, we truncate the myelin basic protein. We virtually divide it to 3 parts, the N-terminal, C-terminal, and the middle. And we truncate it, the C-terminal part, the N-terminal, and the middle part. And as you can see, the protein was still hydrolyzed by the proteasome in all these cases. Moreover, we made some mirror twin of the myelin basic protein, so we reversed all the sequence from C-terminal to N-terminal, and it was still hydrolyzed. And the conclusion was that, the first conclusion was that myelin basic protein contains multiple direct proteasomal signals, and the second, that these signals are not really sequence but composition dependent. To verify this, we analyze the distribution of the amino acids in the myelin basic protein and compare it with the distribution in the human proteome. And you can see the group of outliers. And this is not surprising that in the myelin basic protein there are a lot of basic amino acids like lysine and arginine. And we generate some artificial analog of the myelin basic protein. We took the first 6 amino acids, the most abundant in the myelin basic protein, and generate, as we called it, basic elementary autonomous dogma. So this is the sequence repeated 9 times, fused with DHFR with some unstructured region that helps to proteasome to engage this artificial protein. And you see here that this artificial substrate was hydrolyzed by the proteasome, whereas DHFR itself and DHFR with unstructured region, they both were stable. And when we substituted these basic amino acids, arginine and lysine, to the glutamine, such substrate was stable in cells, suggesting that the basic charge is the main driving force of the ubiquitin-independent proteasomal hydrolysis. So we tested this on the myelin basic protein. You see here we generated some variants where we substituted some arginines and lysines in the sequence of the myelin basic protein to disturb this positive, this basic network on the surface. And vice versa, we generated variants that all lysines were substituted to arginines to increase the basic charge. And this is the native gel. You see here that the most charged variant, this is KR, then wild type, and there's 2 variants with the substitutions of the basic amino acids to the neutral. They are less charged. We tested all of these variants in vitro hydrolysis by the proteasome. And this is the wild type. It is hydrolyzed. KR variant is hydrolyzed even faster. And 2 variants C and RQ that have the less charge, the less basic charge. They are quite stable. So in case of the MVP, the situation is the same, that basic charge is the driving force of the ubiquitin-independent proteasome hydrolysis. And the next step was to realize that what regulatory complex of the proteasome traps the myelin basic protein. There are 2 major complexes, so-called 19S regulatory subparticle and REG-alpha beta or REG-gamma. We use the siRNA to silence all these subunits. You see here REG-alpha, beta, and gamma, and RPT1. This is the most important structural subunit of the 19S regulator. And in case of the myelin basic protein, it was accumulated in case of silencing REG-alpha and REG-gamma. And in contrast to ubiquitin-dependent substrate, ZS green fused with the ubiquitin, it was accumulated only in case of silencing RPT1. denoting that this is only 19S regulator is important for its hydrolysis. We tested in vitro binding of the myelin basic protein with REG-alpha. You see here two variants of the MVP, the wild type and the RQ variant, with the substitutions of the arginine to glutamine. And if the wild type variant had KD around 0.2 micromolar, so deaminated, quasi-deaminated variants of the MVP as at least 10 times worth KD of the interaction with REG-alpha. Next, we analyze what the structural part of the REG-alpha is essential for the interaction with the myelin basic protein. So this is the structure of the REG-alpha. So this is the heptamer, and we notice that REG-alpha contains some flexible and negatively charged loop. You see it here in green. And when we substituted this loop by flexible GS, neutral GS linker, you see here that the hydrolysis of the myelin basic protein was significantly suppressed by this substitution. This is 20S kept with the REG-alpha, and this is hydrolysis of the myelin basic protein, and this is REG-alpha with deleted negatively charged loop. The myelin basic protein was significantly more stable. And the conclusion is that more likely that interaction of the myelin basic protein and REG-alpha is mediated by the complexes of the arginines with negatively charged amino acids like glutamate and aspartate. So finally, we suggest that unfolded tentacles of REG-alpha, as we called it, rectopus, may trap the MVP cooperatively and mediate its ubiquitin-independent hydrolysis. So just to remind you that the proteasome is a major supplier of the peptides that are presented on the MHC class 1 molecules and are essential for the recognition of the cells by the cytotoxic lymphocyte. So that was the conception that probably some changes in the composition of the proteasome inside the oligodendrocytes during MS may somehow be involved in the MS progression. And first, what we tested, if there are any shift of the balance between so-called the proteasome and the immunoproteasome. Immunoproteasome, this is a variant of the proteasome with another catalytic subunits that hydrolyze proteins and generate peptides that are significantly more suitable for the presentation on MHC class 1 molecules. And you see here that the beta-1 is the subunit of the constitutive proteasome, and this is immuno. Same for beta-5, beta-5I. And this is segments of the brain from the SGL mice, from the non-immunized SGL mice, and this is brain segments from the mice developing experimental autoimmune encephalomyelitis. This is the rodent model of the MS. And it's evident that in case of EAE, the balance is shifted from the constitutive proteasome to immunoproteasome, and same for beta-5. So the amount of the beta-5 is significantly elevated in the brains, in all brain segments of the mice with EAE. So we decided to analyze is there any changes in pattern of the hydrolysis of the myelin basic protein by the constitutive proteasome and immunoproteasome. And in order to elucidate this, we used this approach. It is known that the proteasome during each cycle of the peptide bond hydrolysis incorporates one oxygen from the water to the C terminus of the generated peptide. So it is shown here. Using this property of the proteasome, we hydrolyze the myelin basic protein by the constitutive proteasome in water with oxygen with mass 18 and by the proteasome from immunized mice in ordinary water with oxygen 16. Then we mixed these peptides, equimolar generated from equimolar amount of the myelin basic protein, and use classical LC-MS to analyze pattern of the hydrolysis. Of course, all of these peptides were chemically identical, and therefore their retention time and the ionization capability was absolutely the same. Therefore, we were able to compare more than 250 peptides quantitatively in one analysis. And you see here the results. We detected at least 2 peptides, this one and this one, and the amount of these peptides was increased up to 10 times compared during the hydrolysis of the myelin basic protein by Immunoproteasomes compared to the hydrolysis of the myelin basic protein by proteasome from the non-immunized and normal bulb C mice. We used another method with isotopically labeled standard and verified the previous analysis. And here is the time course of the release of the encephalotogenic MVP peptide. You see that the immunoproteasome from immunized mice generate significantly more amounts of the MVP peptide compared to the proteasome from normal mice and non-immunized mice. Importantly, that detected peptides, this one, particularly this one, it is targeted by the cytotoxic lymphocytes, at least in vitro, and very similar peptides were found on the MHC class 1 molecules eluted from the CSF of MS patients. And then finally, our question was that if we know that the beta-1I and beta-5I, both of them are elevated, so what's our target, beta-1 or beta-5? To elucidate this, we stained the brain sections for beta-1I and beta-5I, and you see here that beta-5I was likely be in infiltrating lymphocytes, whereas beta-5I was in the resident brain cells that are surrounding these blood vessels. And we proved this using double staining CD3 with beta-5I and myelin basic protein with beta-1I. And yes, we showed that definitely beta-1I is elevated in oligodendrocytes, whereas beta-5I is smuggling to the CNS with infiltrating cytotoxic lymphocytes. We tested specific inhibitor of beta-1I in vivo, and as you can see, this is the red curve, this untreated mice. The blue one is unspecific inhibitor PS341, also known as bertozumab. There is a little decrease in the MDS, in the main disease score, and when we administrated beta-1I, the suppression of the EA development was more significant. So some conclusions that we suggest. The first one, MVP was shown to form amyloid-like fibrils, and probably free MVP is potentially toxic to these cells. And the first conclusion is that proteasome may regulate the amount of the free MVP, therefore defending cell from some amyloid fibrils that may be formed by the myelin basic protein. And the second, that hydrolysis of the myelin basic protein by the immunoproteasome may contribute to the development of the multiple sclerosis. That's the IFN gamma that is released by infiltrating lymphocytes through the IFN gamma R and STAT1 dependent mechanism, shift the balance from the protosome to the immunoproteasome, and this variant of the immunoproteasome with high amount of the beta-1I generates significantly more peptides that are suitable for presentation in MHC class 1 molecules, and that are in turn recognized by the cytotoxic lymphocytes that attack the oligodendrocytes. And there is a feedback loop, there are more alpha and gamma, more immunoproteasome, and let's have it. So finally, I would like to thank the organizers for this great opportunity, and of course my colleagues, and especially Anna Kudraeva and Ekaterina Kuzina for their outstanding contribution to this study. Thank you very much for your attention.
Thank you very much.